Hello Bruins, and today's screencast is part 2, and we'll talk about stages of mitosis and cytokinesis. Now, when we talk about cell cycle, we're basically saying what is the lifespan of a cell and what happens during its lifespan. So let's start with the G1 phase. The G1 phase is the beginning. And this is where the cell will conduct its regular business, produces proteins, cellular transport, cellular respiration, all the things a cell needs to do. And if the cell is not going to divide, we say it's going to stay in a G0 phase. And there's some cells that don't divide, like the nerve cell, muscle cells. And if the cell is going to divide, then it'll go in a G1 phase. After the G1 phase, they'll go to what's called the S phase. And S phase is the phase where DNA is copied. And after DNA is copied, then the cell will go into the G2 phase, where further organelles are duplicated and it gets ready to divide. So the G1 phase, S phase, G2 phase is what we call the inner phase. When the cell is ready to divide, it goes in the M phase. And the M phase is basically the division of the nucleus. Remember, this is happening in the eukaryotes because eukaryotes have nucleus. And the M phase is further divided in phases: prophase, metaphase, anaphase, telophase. And once after telophase, cytokinesis happens and the cell divides into two. So like I said, cell cycle, the first part is inner phase, the non-dividing phase, and that's broken up into three parts. Mitosis is the part where the nucleus divides, and for the nucleus to divide, it's got to prepare its DNA and make sure all its DNA is packaged properly, and each cell will get the correct number, the correct copy of DNA. So prophase, remember, P prepares chromosomes. Metaphase, M, chromosomes line up in the middle. Anaphase, A, chromosomes pull apart. And telophase, the T, think of it, chromatids in two nuclei. And cytokinesis, cytoplasm divides. Organelles and other cell membrane divides. Let's look at the pictures. Now, when you look under the microscope, it's interesting to know what happens, how DNA is packaged. So during interphase, we say DNA is in the form of chromatin. Chromatin is basically loosely packaged DNA. So here in the cell here, here's the nucleus, chromatin, here's the nuclear membrane. Out in the cytoplasm, you have centrioles, or a region which is called centrosome, a region where spindle fiber is formed. And spindle fiber here is basically a microtubule that's going to help with cell division. So when the cell is in prophase, the chromatin turns into chromosome. And basically it condenses, and under a microscope you could see dark bodies, and that's what chromosome means. The nuclear membrane starts to disappear, spindle fiber starts forming, and right before metaphase, you see that the chromosomes are attached to the spindle fiber by this protein called kinetochore, and kinetochores attach the chromosome to the spindle fiber in the center here called the centromere. Now, once the cells line up at this equator or imaginary equator here, we call that metaphase. Remember metaphase as in the middle. And notice the chromosomes are all attached to the spindle fiber. Then it's ready for anaphase. Here, notice that each sister chromatid, meaning they're all exact copy of each, is going to go to each end of the poles. And the chromosomes are pulled apart, and then when it goes into telophase, the opposite of prophase happens. the nuclear membrane reforms, the chromosome becomes chromatin, the spindle fiber starts breaking down, and here you can start seeing the cytoplasm pinching in. Think of it as a garbage bag with a drawstring, and you pull it and it closes the bag. So it sort of acts like that. Now, just for structural parts, what helps in cell division, you have your spindle fibers, and they're made up of protein called microtubules. And when we say microtubule, think of like hollow tubes. And these microtubules are formed in the area called centrosome or mitotic centers. And if you remember, animal cells have centrioles where plant cells don't. And then here's your chromosome with two sister chromatids attached at the centromere, and they're attached to the spindle fiber by something called kinetochore. So to me, I think of it like a cable car, and the cable car is a chromosome, and the pulley is like the kinetochore, and these lines are the cables. All right, let's go in more in detail. Interphase, remember, includes G1, S, G2. It's an in-between phase of cell division, or should I say nuclear division. Cells spend most of their time in interphase, and when you look under the microscope, you can see the nucleolus. If you remember, that's where ribosomes are made, or RNA is made. And here is the nucleus, and then here you see chromatin. Now in prophase, we see, when you look under the microscope, you see the chromosomes. They're distinct. They kind of have this wormy kind of look to it. And here we call the chromatin chromosomes. Centriole starts to appear. You don't see that in the microscope, but a spindle fiber starts to form, and the nuclear membrane disappears. So here's a whitefish embryo called a blastula, and here, as you can see, the chromosomes are distinct versus, let's see, here in this cell you don't see the chromosomes. Now metaphase, remember, M for middle, where chromosomes are attached to the spindle fiber, and this is what you will see under the microscope. And here's a picture of white fish embryo. As you can see, the chromosomes are lined up. And then anaphase, centromeres separate. The spindle fibers, there's a pulling effect, and also the chromatids are like walking and at the same time being pulled to opposite ends. So each end of the cell will have a sister chromatid. And this is how it looks in the whitefish embryo. And telophase, think of two nuclei. You see starting to see two nuclei. It's opposite of prophase. So here the nuclear membrane will start to reform. Chromosomes will spread out and become chromatin. Centrioles and spindle fibers will disassemble. And then here you can see it in the whitefish embryo. And then cytokinesis kind of starts happening at the same time as telophase. The cytoplasm starts splitting, and here you see what we call a cleavage furrow, where the cell starts pinching in. And in plant cells, since plant cells have cell wall, you don't see pinching in, but there's vesicles that'll deposit cellulose, and then the cellulose basically merge and become a cell plate, and eventually dividing the cell into two. So here's a difference between animal cells and plant cells. So animal cells will have cleavage furrow, plant cells will have cell plate. Okay, so here, I'll put this on Blackboard and you could review these diagrams. Let's look at these pictures. This is an animal cell where we use special fluorescent dyes, and the DNA is in blue and the spindle fibers in yellow. So here we have a cell in interphase, and now as you can see, the chromatin chromatin is condensing and you see distinct chromosomes. So this is definitely in prophase. And as you can see now, the chromosomes are moving towards the middle. And until when they're finally at the middle, we say they are in metaphase. And here, as you can see, here's the spindle fiber, chromosomes in the middle. And now here you see the spindle fibers are pulling the chromosomes apart, anaphase. And here you start seeing telophase, start seeing the cleavage furrow here. And we also will look at the onion root tip. So here's a region where you would see a lot of cell division, and we'll also look at the whitefish embryo, because in these two we have a lot of cell division. Now think about, why do you have cell division? Here we're having growth happening, and here we have cell development. Here's a microscope picture of onions, and you can see the rectangular cells, and these cells are in interphase, a chromatin, and there's a nucleolus. Now here you can see distinct chromosomes. You don't see the nucleolus. Here the chromosomes are moving towards the end of prophase, and when they come to the middle, that's when we say it is in metaphase. Here's example of metaphase, and here's telophase, and here's the cell plate forming. And this, notice, is interphase. Here's anaphase, chromosomes are moving apart, early anaphase, late anaphase. Here's telophase, and again you can see that cell plate. So here's telophase, anaphase, interphase. Notice interphase, you don't see distinct chromosomes, it's a chromatin, and here's a nucleolus. So when you see a nucleolus, you definitely know it's an interphase. Here's very late telophase, here's a cell plate. So we're going to do a lab where we're going to look at cells, and we're going to count how many of these cells are in prophase, how many are in metaphase, anaphase, and telophase, and figure out the percentage of cells spend in what phase, and figure out the time they spend during mitosis. So look for that lab on Blackboard.